Hi, my name is Srijani Datta, and my project is titled Sepsis Survivor. So, a little background on my project. Sepsis is a medical emergency, and it is the body's extreme response to an infection. It affects about 1.7 million Americans per year, and unfortunately, 270,000 people pass away from it. What's even worse is that even those who survive, out of them, 50% are burdened with long term disabilities, including cognitive impairment, cardiovascular complications, as well as sepsis induced myopathy. So, on the right, there are some symptoms of adult sepsis. This is sometimes some of the things that you'll see if somebody is having sepsis or their body is reacting in that way or starting to react like that. So, what is sepsis and muscle wasting? So, sepsis induced myopathy is inefficient muscle regeneration. This means that the body is not producing or reproducing muscle as fast as it's losing it. And essentially, your muscle mass is drastically decreasing. This lowers your quality of life, and you are unable to do basic tasks. You're unable to do basic tasks. So, what's supposed to happen is after any trauma or injury, muscle stem cells, or satellite cells as they're called, are supposed to proliferate into myoblasts. These myoblasts then differentiate into myocytes, which are muscle cells, and then the myocytes fuse together to form myofibers, which where the myofibers are the muscle cells the myocytes working together to function as one cell. So myofibers are what we ideally want for muscle formation. But unfortunately, with sepsis, trauma, this does not happen. For whatever reason, skeletal muscle is very negatively affected during and after sepsis, and there's no available treatment for this muscle wasting. So, how this project was conducted was that CLP-DCS was performed on mice to mimic sepsis progression through a human being. So, CLP is cecal ligation and puncture, which is basically where a mouse's cecum is punctured, fecal matter is let out to give it infection, the sepsis, and then as they recover, they go through DCS, which is daily chronic stress, to mimic a human being in a hospital bed, just to mimic like how human does when they go through sepsis. And the genetic material was collected from this mouse, from the hind muscle in the leg, and it was isolated, and satellite cells were isolated and turned into RNA. And all of this was done before I was involved in the project. My involvement started when the raw counts from the RNA sequencing came in an Excel sheet, and that is the Excel sheet that I got. From that, I filtered it so that the top 20 genes of change and the top 20 negative changes from control compared to day 28. So if we look at this diagram down here, the genes were sequenced, RNA was sequenced at 3 different time points, and then control was sequenced as well. So at day 4 sepsis, day 4 post sepsis, day 8 post sepsis, and day 28 post sepsis. So I filtered based on changes of control compared to day 28. And then I also found a literature review which found genes that have been linked to muscle hypertrophy, which is the overproduction of muscle. And I chose 5 genes from this literature review to also look at in addition to the 40 genes. So I looked at 45 genes total. My hypotheses were that one, that there will be genes that code for proteins that are involved in muscle regeneration that have a lower expression value at the day 28 time point compared to the control. And then my second hypothesis is that there will be genes that will stop being expressed completely at the day 28 time point, and then there will be genes that have already been deemed significant, and this is reflected in the RNA sequencing data. So for my results and discussion, I found 14 genes that contributed to satellite cell differentiation or proliferation, and the genes are listed in alphabetical order. So first up, we have ACVR2B, and this gene is significant because the gene has been linked to muscle hypertrophy. So the expression going down during sepsis recovery is not too promising. It does start to come back up at day 28, but we don't know exactly like how that plays out or if it ever reaches control again. So this is a gene that could be enhanced as a potential treatment option. Then we have ACTC1, which causes muscle diseases. So the increased expression increases chances for muscle disease. So this is a gene that should be inhibited for a treatment option to prevent muscle diseases and increase muscle diseases post-sepsis. Then we have AKT1, And this pathway has been very concretely proved to promote myoblast proliferation. So it is very concerning that at day 4 and day 8, the expression of this gene disappears completely. We completely lose this pathway of myoblast proliferation. So the treatment option right away is bringing this pathway back day 4 and day 8 and keeping it consistently there throughout recovery, and not just having it like blip back occasionally, so that we have consistent muscle regeneration. Then we have CARM1. This one is very similar to AKT1, as CARM1 has been concretely proved to regulate signaling for skeletal muscle, and there's been multiple studies that show if you don't have CARM1, there's reduced muscle mass. So this is one that we want to really push, and the fact that it's not there at day 8 and day 28 is alarming, and we want it to be there so that we have muscle mass and we have regeneration. So this is one we want to enhance. Then there's EIF4A2, and this gene has been in the literature has been shown to increase levels mean more myofiber formation. We want myofiber formation, so this looks promising that at day 8, day 28 post sepsis, we do have higher levels than control. So we want a potential pathway to further increase so that more muscle regenerates. And then we have firm T2, and for proper wound healing, we want there should be elevated levels of firm T2, so that since there are no levels, like it's not expressed at all post sepsis, this is very concerning for wound healing, and sepsis patients have been shown to have slow wound healing or very poor wound healing. And so this is one we want to enhance so that wound healing gets better, and that comes with regeneration. If your muscle isn't as hurt, it can better regenerate. Excuse me. Then we have FHL1, and this gene helps maintain normal cellular structure and formation and function. So this pattern lends itself very clearly to the idea that only some genes are hurt by sepsis at day 4 and day 8. So enhancing during that period, or potentially lowering the amount that we're pushing this for treatment. Then we have FST. This plays a role in bone mass, and so the pattern very clearly follows the lean mass change, which I thought was very interesting. And also it's a gene that we can enhance so that it doesn't decline and we have consistent bone mass. Then we have MEF2D, and this is a gene that also contributes to myoblast proliferation, a very similar pattern to the AKT1, where there's just not expressed at all post sepsis. So we're completely losing this pathway. So we really want to enhance this one. Then there's MSTN, and increased expression of this gene is linked to muscle wasting. So we want to decrease it, and we want to inhibit this expression. So this was very interesting that I found. Then we have SKI, or SKI, and reduced expression of SKI decreases cell differentiation. So in the pattern, fact that it's consistently decreasing from day 4 to day 28 compared to control is really concerning. We want SKI to be expressed so that we have increased cell differentiation. So we want to inhibit this pathway, or not inhibit, enhance this pathway. And then we have THRA. THRA expression going down and not being expressed is bad for muscle regeneration, as it maintains satellite cells. We want more satellite cells. So we want to enhance this pathway and bring it back. And then PDLIM5, it also essential for differentiation. This pattern expression is kind of crazy, so we want to look into why this happens. I thought it was very interesting. Pathway should definitely be further explored. And lastly, we have WNK1, or WNK1. This pathway is another one that is related to hypertrophy. In the absence of it actually reverses myotube atrophy. So we want to push this pathway to try and reverse the atrophy that happens. So in conclusion, the first hypothesis was proven true by 2 genes that had higher expression at day 28 than control. Second hypothesis was also proved true. There were many genes that were not expressed at day 28. And the third hypothesis was partially proved true, as there was a list of genes that were contributing to muscle hypertrophy, but the expression level was not as drastic as I thought it was going to be for how the gene changes. For future work, we ought to look at each of the specific genes mentioned for how we can inhibit and enhance it. And we should also look at all the genes in the spreadsheet and to individually analyze them. I relied on many journal articles as I executed this project. The resources I read really informed my understanding of current knowledge in this field, and all images are by me, the author, unless otherwise noted. This research is a gateway to developing an effective treatment for sepsis-associated muscle atrophy, which would be life-altering for millions of survivors across the globe.